All right, ladies and gentlemen, here is your first part of DNA structure and replication. We're pretty much just going to go over what DNA is made up of, what the point of it is, and how it can be replicated and why. Okay, so you guys should have a note template that you guys are going to be jotting down everything you need to. So if you need to pause it, do so, because sometimes we go a little bit fast just to get the video under time, but you still might need a little extra for yourself. So this is a little cartoon on DNA, so you can read it for yourself, see what you think, and then we'll go actually into the real structure and what it looks like. So there's three parts to a nucleotide. Before we talk about what the three parts are, let's talk about what a nucleotide is. All it is, it's the building block of the DNA. This is what makes up all of the deoxyribonucleic acid. So that's what DNA stands for. So let's look at these parts. Part one is there's a phosphate group. Part two, there is a deoxyribose, which is a pentose sugar. Pentose just means 5, like pentagon. And then there's the nitrogen bases, and this is what we're going to focus on most of the time, because the nitrogen bases is what ends up coding for certain proteins that we're going to see in the next couple videos. So just keep these nitrogen bases in the back of your mind. We're not going to worry about these words. Actually, we're just going to get rid of that altogether. What I want you guys to really focus on here are these two nitrogen bases, guanine and cytosine. Guanine and cytosine. These guys always are together, and then the adenine and thymine. So the reason why I paired these two together and then these two together is when we're having our DNA being created, this G, we abbreviate CAT, these are the ones that always pair together, and these ones are too, and we call them complementary bases. And just complementary means that they're always paired up together, like that word right there. So here's the chemical structure of it. It looks a little bit complicated right there, but I'll just highlight a few of the main parts. Here's our phosphate, which is PO4. So this is our phosphate part. This is our 5-carbon sugar. That's the pentose, 5 carbons. And then these guys in here are our bases. So really what this kind of looks like is almost a ladder shape, where we have our phosphate and our sugar alternating, and then we have our little ladder rungs here, which would be our G's, C's, A's, and T's. But we'll see this in a few minutes. So here's a better picture in terms of, it's not as chemistry, it's just kind of a little bit more symbolic of ways to remember it. So here's your 4 different bases. So here they are. And here's your sugars here. And then your phosphates that alternate. Okay, so this is the general structure: phosphate, sugar, and bases. So we call this the backbone, the phosphate sugar backbone. And like I was saying, this would be like the ladder sides. If you're going up a ladder, it's the phosphate sugar. And it's twisted into a structure that looks like this, which is called a helix. And what connects these bases here is a bond called a hydrogen bond. You'll see this in Chemistry 11, and then you'll also see it in Biology 12. And what is this DNA? The whole point of it is when you have large sections of DNA, it makes up a thing called a gene. And all that a gene is it determines which proteins are going to be made in the body. Okay? So what's the purpose of replication? So we know the structure of DNA. We're going to be doing some stuff with that in class. So if you're a little bit fuzzy on it, just bear with me. But replication means to duplicate or to make more of. And the whole purpose of that is if you get injured. If you get injured or you're growing, and some of your skin cells are broken, you need new cells, but you don't want them to be different, because all of your skin cells, you need the same DNA so you can make the same protein, so you can have all the same genetic code. So it needs to make it perfect. And the way that it does that is through replication. And what happens here is that if we have our DNA strand that looks like this, what happens is it gets opened up and we copy each side here. We'll copy this side, and then we'll also copy this side. So it looks like this, and this is going to look real confusing, but we'll break it down into some steps and we'll work on the concepts in class here. But as you can see, here's our DNA strand that's been unwound, so it looks like a ladder. Half of it goes this way, half of it goes this way, and what this is all this green parts, these are all the different bases and on the same side. So what happens is that new stuff is added in a complementary fashion. So if we have G, C, A, T, what happens is this is going to be added. G's always go with C's, and A's always go with T. So we know exactly what's going to go there. All right, let's look at the process. So step one, we have something called an enzyme, which is a type of protein that speed up reactions. This one's called helicase. So write this down, pause it if you need to make sure you get it all down. The helicase is going to break apart this helix. And then what happens, like I was saying, DNA nucleotides, which was the phosphate, sugar, base, are going to be added. So as soon as it's broken apart, new bases are going to be added right when it breaks. And they're positioned by another enzyme which is called DNA polymerase. So the helicase breaks it, and then the DNA polymerase pretty much just goes behind it and places it in certain areas. And then finally, a third enzyme, where are we here? DNA ligase is going to fix any breaks. So if they happen to, it's kind of like spell check in your computer. You're typing away, typing away, typing away, and then spell check finally will check it for any errors. This is what DNA ligase does. So if anything is added incorrectly, or if there's a gap, this will fill in the gaps and make sure that it's all perfectly sealed. All right, so hopefully you might have to go back and pause it and jot it all down. But this is what you need to know. 3 parts of a nucleotide. So we have our sugar, our phosphate, and then our nitrogen bases. You need to know the 4 types of complementary bases. So G's goes with C's, A's and T's. I remember this just because G's and C's both kind of have the round. A's and T's are kind of both straight lines, and so they match up together. And then finally, make sure you know the steps of DNA replication. More specifically, make sure you know the 3 types of enzymes that we talked about. The helicase, the ligase, and the polymerase. Okay, so if you have questions, jot them down. We'll be going in lots and lots and lots and lots of detail in order to get this into your heads. Have yourselves a wonderful night.